looking forward to this. And I guess I'll just leave the intro short, Heather. I'll let you do the rest of the intros, but I'm really looking forward to this. Thanks for coming.
Cool. Thanks for having me. So hi, everyone. I'm going to give a talk today on a study I've been working on for many years. It's called the Wells and Enteric Disease Transmission Trial, or the WET trial. And it's a randomized control trial. But first, I'm going to talk a little bit about my background on how I got into WASH, because I was asked that I should probably do that. So I added a couple slides in a few minutes ago, and I'll maybe just go through my background so you guys know where I'm coming from. Also, before I forget all the lovely people that are listed on this slide, this project had many co-investigators and collaborators. So I'd like to acknowledge all those co-authors at the beginning, so I don't forget them at the end. So let me see, what can I tell you about myself? So I have these slides that I often give to students when I'm giving classes on my background and how I got to where I am in my career. So I did my Bachelor's of Engineering, Environmental Engineering, and in my master's at Dalhousie University. And I focused my master's research all on drinking water distribution systems. And all the work I did on my undergrad and master's, I was really interested in drinking water treatment. And from there, I decided I didn't want to treat water for people who literally flush it down the toilet, and thought that I would really like to make a contribution overseas. So what I did was apply for multiple internships. I was unfortunately unsuccessful at getting any of those to go and do work overseas. And instead, I worked for an engineering company for a few months, then took all the money I made, bought a plane ticket, and went overseas and traveled through Southeast Asia. And I had been awarded an Enteric PhD scholarship while I was gone, and I deferred the starting point as long as I possibly could, and then came back and started a PhD at the University of Guelph and developed a project to look at point of use water treatment in Cambodia. So that was my first, I guess, stint in the WASH sector, so to speak. And I left academia in, when did I graduate? 2010, and worked for UNICEF for a couple of years in Mali and Madagascar. And then slowly made my way back to academia. I got really interested in the public health implications of the work we were doing with UNICEF, and worked with quite a few academics who were running epidemiological studies. And so I ended up taking a postdoctoral fellowship with the Public Health Agency of Canada, where I worked on quantifying the burden of disease due to drinking water. And so that was one of the headlines of the National Post when our research articles came out. There, the study reveals hidden dangers of tap water, millions of Canadians drinking from substandard sources, which isn't exactly the case. They kind of sensationalized the results, but that was a brief snapshot of that work. I then did some postdoctoral fellowship work again at the University of Guelph and McMaster before I got a position in the United States in a college of public health. So I left engineering and moved into kind of environmental health. And I've worked mostly on waterborne disease transmission, and I've done a range of projects in North America and also overseas, and have now made my way back to Canada. And I'm at the University of Guelph in the Department of Pathobiology, which is actually in the vet school. And they hired me to do kind of interdisciplinary One Health research. So I still work a lot in the water and sanitation space, but also with more of kind of a One Health lens. So that's a bit about me. I think I'll get going into my actual presentation. So the trial has been a work in progress for, I guess, probably 7 years now, and is still ongoing. So I have some preliminary results to show you, but I'm going to start by giving you some background on groundwater and pathogens and enteric disease, and then go into the study and some of the preliminary results we have to date. So with that, globally, a lot of people around the world rely on groundwater for drinking. And we tried to crudely estimate in one of our papers. And so we came up with, in Canada, around 20% of the population relies on groundwater for drinking. In the US, it's up to 43% of the population. South and Latin America, we estimate around 23% or 150 million users, people use groundwater. In Europe, the numbers are a bit more wishy-washy, anywhere between 27 and 67%. In Asia, 23 to 27%, or 1 to 1.2 billion, rely on groundwater. Australia, around 12% of the population. And then the African continent, a large proportion, nearly 80% of the population, relies on groundwater for drinking. And so in total, we estimate that around a third of the world's population rely on groundwater for daily consumption. And groundwater is often chosen as a kind of a more desirable source of water compared to surface water that can be more easily contaminated. However, what's kind of poorly understood and is a bit of a misnomer is that groundwater can be free of pathogens or microorganisms. And generally, people believe that the earth naturally kind of filters the water that makes its way into groundwater and makes it free of pathogens, but that's just not the case. We now know that groundwater can be a significant source of pathogens, but that subsurface pathogen occurrence, distribution, and movement is still poorly understood. We do have enough evidence to suggest that contaminated groundwater is a significant source of gastrointestinal infections in both developed and developing regions of the world. And some examples of how these waters become contaminated include kind of septic systems, leakage, agricultural activities. They can be some of the sources of those contaminants. So for the purpose of my talk today, when I talk about AGI, I mean acute gastrointestinal illness. And that definition we're using is about 3 or more loose stools in 24-hour period, or any vomiting, excluding those with chronic conditions or concurrent symptoms of coughing, sneezing, sore throat, or runny nose. There's many different definitions that exist, but this is maybe a more classic definition, and that's what I'm going to rely on today. So how are wells contaminated? I talked about it a little bit before, but private wells in particular, which is going to be the focus of my talk today, can be contaminated by surface contamination. So around the wellhead where the well is actually constructed, it can happen through the subsurface, through leaking septic systems, or maybe in developing countries, pit latrines and other sources of fecal material. Livestock storage waste, for example, fertilization activities, manure application, and other animal operations. So in 2015, colleagues and I produced a review looking at the epidemiological evidence that groundwater can contribute to global enteric disease. And our review identified 649 published groundwater outbreaks globally, and these are mostly in public water systems. But what we found is that there are numerous papers out there that identified pathogens in groundwater, either during an outbreak or actually just under general surveillance during non-outbreak conditions. And so we found 56 studies that found viruses such as norovirus, enterovirus, adenovirus, and hepatitis A virus. For those who aren't that familiar, these are human viruses, mostly spread from humans to humans. Bacteria, there was 26 studies that found Salmonella, Campylobacter, and pathogenic E. coli. These are zoonotic organisms that can be transmitted between animals and humans. And then protozoa, 10 studies have found, at that point, had found Cryptosporidium or Giardia in groundwater supplies. And these, again, are also zoonotic pathogens. But what I want to point out, particularly for groundwater, outbreaks are really only the tip of the iceberg of what we find in terms of disease. And why is that? Well, it's because not all illnesses are actually linked to an outbreak, which have better surveillance and what we pick up in our surveillance system. A lot of illnesses are endemic or sporadic disease. So they're kind of low-lying in the population, they're isolated, and they go underreported or underdiagnosed. And I'll get to that in just a second. So what do I mean by underdiagnosis or underreporting? This is a triangle that was shared with me when I worked at the Public Health Agency of Canada, and it really illustrates how we lose all of these cases in our surveillance system. So at the bottom of the triangle, we have what is the total estimate of illness in the population. So this is what the true illness is, but at the top of the triangle is actually what we actually detect in our surveillance system, which is the tip of that iceberg. So moving from the bottom to the top, if we talk about underdiagnosis, I don't know if I can see your hands on the screen, but if I were to ask all of you, last time you had a bout of diarrhea, did you go to the doctor? So how many people went to the doctor? Is there any hands? No hands, right? Nobody went to the doctor. So we lost you all to the surveillance system. You guys are not picked up in surveillance at all. If you've ever had a bout of diarrhea that was so bad you went to the doctor, How often did the doctor even ask you to submit a stool sample? And if they did, were you too grossed out to even submit it? So how often did that specimen actually get submitted? And then if that specimen makes it to the lab and the doctor ordered the right lab test, did it actually get tested for that pathogen of interest? And then how sensitive are our laboratory tests? So we lose all of these cases of enteric disease due to underdiagnosis. We also then lose them due to underreporting. So how often are laboratory confirmed cases actually reported back to the doctor, for example, and then how often does that case make its way into our surveillance system? So this will be important as we kind of go along and talk about enteric disease. So then how do we actually measure this sporadic or endemic cases of illness? Well, we rely a lot on epidemiological studies, and in terms of drinking water studies that have been done, we've got randomized control trials, community intervention trials, case control studies, and ecological studies. There's also a method called quantitative microbial risk assessment, which relies on data on the presence of pathogens in, say, a water source, and then the probability that you're going to ingest that organism through drinking water, for example, and what's the likelihood that you're going to become sick. So these are the different ways that we try to measure sporadic endemic disease, particularly in drinking water. And for groundwater, I'll go through what we know from the existing epidemiological studies. So for randomized controlled trials, this is the gold standard of epidemiology. So for those who aren't that familiar, it's essentially like a drug trial. So what we do is we compare illness rates between two randomly chosen groups of people. And so for a drinking water trial, the control households would drink tap water, and the intervention households are then equipped with some sort of water treatment system. And the idea is that these two groups are nearly identical with one another because they've been randomly selected. And the only difference between them is their drinking water source. And so if we see a difference in illness, we would like to attribute that difference in illness to the intervention or the drinking water treatment. So if people got less ill in the intervention group, we would assume that that was because of the intervention and not because of other causes. So 5 full-scale randomized control trials have been conducted on large-scale drinking water systems worldwide. 2 were done in Canada back in the late 90s by Pierre Paiement in Montreal. 2 have been done in the US by a researcher named Jack Colford, and 1 has been done in Australia by colleagues down at Monash University. And these studies have been all done on public drinking water systems that were not groundwater systems, and they were all treated water supplies. And so why haven't more of these been done? Well, they're really costly and time consuming, and so therefore our data is really limited. And importantly, no one has done one looking at illness attributable to untreated groundwater supplies or private wells. So this has not been done to date. The next best thing, if we look at kind of the quality of evidence for epidemiological studies, is a community intervention trial. And so it's very similar to the randomized control trial, except it's done at the community level. So the treatment happens at like a community well, as opposed to in your house. And so one study has been conducted by my colleague, Mark Borkart in Wisconsin. It was done on 14 municipal undisinfected groundwater systems. There were 8 intervention communities and 6 controls, and they used ultraviolet light at the wellhead. And they saw a 6 to 22% increase in acute gastrointestinal illnesses due to when they found viruses in the tap water. And they also found a 1 to 4% attributable illness due to intrusion in the distribution system. So they think that pathogens were making their way in the distribution system, which is what was causing people to become ill. And they just finished a similar trial in Minnesota about, I want to say, a year or 2 ago, and those results are still pending publication. The next kind of study are case control studies. So the study begins by selecting subjects based on their disease or no disease. So we would have people who have gastrointestinal illness and are matched to someone who maybe has like a broken leg. And then you look back to see what their exposure is and whether or not one might have led to disease. So disease in our case was acute gastrointestinal illness, and then we would trace back to exposure to untreated well water. And to date, there's been 4 prospective case control studies of children in the US. So they've looked at cases of AGI or illness from a specific pathogen, and untreated groundwater was an exposure. And so one was done in Wisconsin by Mark Borchardt, and they found 11% of cases of diarrhea were attributable to well water that was contained fecal enterococci. Another was done by my colleague, who's also a co-investigator on my study, Dr. Donna Deno in Washington State, where they showed infection with salmonella in children was associated with well water consumption. Another one was done by Katie Fullerton from the CDC in the United States, and they looked at children aged 0 to 6 months, and they found that they were at risk of Campylobacter if they drank well water. And then another one was done in the US, I think it was in the Atlanta area, if I recall correctly, and they found increased risk of AGI from well water compared to surface water. Lastly, there's these ecological studies, which are kind of the lowest level of evidence we have in terms of epidemiological studies. They measure more than one variable at a group level, usually defined kind of spatially or geographically or temporally. And so what they'll do is kind of look at exposures in a geographical area. And these exposures are averaged within the population and then analyzed using a variety of statistical approaches. So you'll look at drinking water source or rainfall or septic tank density, and then your illness outcome. And so do you think there's an association at a group level between these different things? And so in our review at the time, we had recovered 5 studies, and 4 of the 5 showed a link between consumption of groundwater and acute gastrointestinal illness. The last method I'll talk about briefly is this quantitative microbial risk assessment. So this is not an epidemiological study. This is a modeling procedure. And essentially the way QMRA works is it determines the likelihood and extent of human health effects following exposure to a microbial pathogen. So it doesn't need to be used just for drinking water. It can be any exposure to any pathogen. But the key inputs are pathogen-specific occurrence data. So for us, we would need the amount of pathogen, like the actual concentration of pathogen in drinking water or in groundwater. Then we want to know what the infectivity of these pathogens are, and we have to estimate the probability of somebody actually consuming water containing that pathogen. So how much do people actually drink of this water, and what's the concentration of that water, of pathogen in that water? And typically for drinking water, the result comes out of a risk of illness or infection, and the acceptable risk is generally 1 in 10,000. The advantage of this approach is it's much less costly and time consuming than epidemiological studies. And actually, there's now been a couple of instances where people have compared epidemiological data to QMRA and they have come quite close. So there were, at the time, I'm pretty sure there's lots more of these that have been done since my review, but there were several done on Cryptosporidium, E. coli 0157, Giardia, and rotavirus. So they're very pathogen specific. So if you want to do a QMRA for all pathogens, you have to build a mathematical model for every single individual pathogen. So more recently, we just concluded another systematic review with some colleagues in the United States, looking at the burden of disease from contaminated drinking water in countries with reportedly high access to safely managed drinking water per the Joint Monitoring Program. And that's currently under review. But we found that private wells were a significant gap in our knowledge and what the burden of disease was associated to private wells, and what are the groundwater contaminants of emerging concern, and pathogens was one of them. So we've done kind of 2 concurrent reviews. And what we know is there's still increasing evidence of microbial well water contamination, and 2 new studies that came out from my colleagues in Minnesota and Wisconsin have showed pretty striking results. Like, people generally think that groundwater is free of Cryptosporidium, but in Minnesota, they found 40% of wells were positive of Cryptosporidium. And the belief was that because it's so large in size that it could not actually be transported to groundwater, but that is absolutely not the case. In Wisconsin, they found that 21% of wells were positive for human bacteroides, which is a human sewage marker, meaning those wells were being impacted by septic waste or wastewater of some sort. And 15% of the wells were positive for rotavirus. And people often will tell me that don't know much about groundwater that even deep wells are never contaminated and they're always well protected, but this is just not the case. And so just one example, I've got numerous, but one is that there was a norovirus outbreak traced back to a deep well as deep as 85 meters in 2011. And that caused a huge sickness of people that were consuming water from this particular restaurant. So what is the burden of disease attributed to private wells? Well, there's no robust estimates that exist, and there's very few epi studies, as you can see. So in 2016, when I was at the Public Health Agency of Canada, when I did that burden of disease work, we did come up with a crude estimate using quantitative microbial risk assessment. And I realized I have a typo in that. That's supposed to be 70,000. There's an extra zero. There were 70,000 cases of AGI in Canada attributed to Campylobacter, Cryptosporidium, E. coli, Giardia, and norovirus. And we extrapolated those numbers to the US because I was in the US when I started working on this study. And crudely, that works out to around 1.3 million cases a year due to untreated groundwater, which is pretty sizable when you think about it. But we actually don't know. These are estimates based off of what we can do with the available data. So I really wanted to know, what is the burden of disease attributable to private well water? And so I spent several years trying to get this randomized controlled trial funded that I'm going to talk to you about today. So it's the WET trial. And that's our website. So if you want to learn about it, you can go there. It's www.wettrial.org. And it's the first randomized controlled trial of its kind globally. And what we're trying to do is see if drinking untreated well water causes gastrointestinal illnesses or respiratory illnesses. And if treating it with ultraviolet light will reduce that illness. So we're comparing people with a UV water treatment device to a placebo device. So the identical device, but it is inactive. And we're focused on children under the age of 5 in rural Pennsylvania. So the aims of the study are one, to quantify disease reduction, primarily acute gastrointestinal illnesses, but we're looking also at respiratory illnesses. And so we're collecting self-reported diarrhea and respiratory symptoms from the parents of these young children. The second aim is to compare child stool and saliva samples between control and UV groups. So we're looking at the pathogens in child stool samples and seeing if we see a difference in what bugs these kids are infected with, depending on whether they drink treated or untreated drinking water. And then we're also going to compare the pathogens in the control well water, so those that are untreated drinking water, with what pathogens we find in child stool or saliva samples, because we're also collecting saliva samples. This work has been generously funded by the National Institutes of Health, and Trojan UV, or VIQA specifically, has donated all of the devices for the study. So why Pennsylvania? Well, I was working in Pennsylvania, so it was highly convenient, but also Pennsylvania just happens to be an ideal state to conduct this work because it has the second highest number of private wells in the country. So it's really high density of private wells. So there's actually in some places, greater than 90 domestic wells per square kilometer. You can see in certain portions of the state. And in order to actually recruit enough people for this study, you want to do it in a densely populated area. So what are the eligibility requirements to be part of the study? Well, the household has to rely on a private well and have no water treatment that provides microbial treatment of well water. So we're allowing softeners and some other systems in, but anything designed for pathogen removal is ineligible. They have to reside in southeastern Pennsylvania. They have to have a child that is 5 years old or younger by the end of our study period, which means we're actually recruiting kids that are 3 years old all the way up to just under 4. They have to drink at least 1 cup of well water a day that's untreated, and they can't have any underlying health conditions that could impact the intervention, or they can't be like significantly immunocompromised. So we are recruiting 908 children under the age of 5. Half of them will be assigned a treatment UV device, and this is a picture of one that's been installed in a household. and the other half will be assigned a control or sham device, which looks identical to that device there. It's just not actively working. These are whole home water treatment systems, so they're at the entry point to the home. The children are followed for 1 year with weekly text messages. And so basically the household index parent gets a text message that says, did your child have any of these symptoms in the last week? And they reply yes or no. If it's a yes, they then proceed to a more detailed illness questionnaire where we capture information about the illness itself, as well as other exposures that they could have had that might have led to that illness and not necessarily the well water. And so we're collecting data on gastrointestinal and respiratory symptoms and comparing between the two groups. We also have baseline, midpoint, and exit questionnaires where we collect demographic information on other risk factors that could cause illness in the kids. So like daycare attendance, food consumption, changes in their water consumption, if they installed a new water treatment system that we didn't know about, those kinds of things. We also have an installation questionnaire that our UV device installer completes. It's pretty detailed. He checks the entire house to make sure that they don't have other treatment systems in place. They look at observational data on the septic system, land use, anything out of the ordinary that could be affecting the intervention. And then we also collect weather data. We have a sub-cohort of households that submit samples. So we don't have enough money to have 908 families submit samples. So 180 households are submitting samples and they're randomly selected. So they will collect stool samples and we will collect water samples from their home at the same time. And we collect them in the absence of symptoms. So we do them when we think they're asymptomatic and also in the presence of symptoms. And then we're also collecting monthly saliva swabs to try a new method for enteric pathogen disease detection using saliva swabs instead of stool samples. For our water sample collection, so we've had a pilot study of 28 homes that was completed, and in the full study, we have 180 homes. We collect a 1 liter grab sample from all the wells, untreated well water sample. And we collect an 800 liter ultrafilter sample. And so the 1 liter grab samples are sent to Temple in Pennsylvania, and the 800 liter ultrafilter samples are sent to the USDA, USGS in Wisconsin. For the grab samples, we take them back to Temple and we analyze them for total coliforms, E. coli, enterococci using membrane filtration in duplicate. And then we also do pH, temperature, conductivity, alkalinity, hardness, iron, and manganese, because they're all factors that could affect the performance of the UV device. For the stool and ultrafilter samples, so we collect child stool samples and then paired well water samples. And we collect these 800 liter ultrafilter samples. Basically, we filter 800 liters through an ultrafilter. I guess it's like a membrane filter essentially in the field, and it essentially concentrates that sample down so we can analyze for different pathogens. And so the ultrafilters are sent to Wisconsin along with the stool samples, and they're analyzed using qPCR or qRT-PCR, depending on the microbial targets. And we're analyzing for all of these different organisms. So we're doing noroviruses, human adenovirus, enterovirus, hepatitis, rotavirus A and C, diarrhegenic E. coli, Salmonella, Shigella, Campylobacter, Giardia, Cryptosporidium, and Shiga toxin-producing bacteria. We're also analyzing stool samples for SARS-CoV-2 to see if kids have been recently exposed to SARS-CoV-2. We also did the water samples for our pilot, but none of them were positive for SARS-CoV-2, so we stopped analyzing that. And also the water is analyzed for human sewage markers, so we know if their water is impacted by human sewage or septic systems. The saliva samples are collected monthly, so there's 12 saliva samples per child. They're sent to Temple where we vortex them, and then they will be going to the US EPA, who has developed methods for looking for specific enteric pathogens using an antigen approach. So they'll look for noroviruses, Campylobacter, and Cryptosporidium. The incentives for the study are that every household gets a free whole home UV water treatment device, which is valued around $1,000 installed US. And even those that are on the sham device will have an active device at the end of the study. Households receive up to $100 in gift cards for timely response to text messages and also for stool samples. Everyone's name is entered into a draw to win a family trip to Hershey Park or Dutch Wonderland. It's kind of like our Canada's Wonderland for kids. And so the more they respond to the text messages, the more entries they get into the draw. And then water testing is for some of the subset participants. They get the benefits of a really detailed water quality report. What do our participants need to do? They need to allow us to install a UV device, complete these different questionnaires, respond to our weekly yes/no text messages, respond to an illness questionnaire when the child is sick, which we estimate around 5 to 6 times a year on average, submit stool or saliva samples if they're in our subset, and allow us to collect untreated water samples from outside their home or in their basement. So some of the strengths of our study is that it's a triple blinded trial, meaning that the family doesn't know if they're on the device. They don't know if they have a true or active device. Our installer doesn't know if he's installing a true or active device. And we don't know who's on a true or active device. We have a whole home device versus point of use. So in the past, most of the studies have been done with, actually all of the studies have been done with a treatment system just at the kitchen tap. So it only treats the water in the kitchen, but doesn't treat all the water in the home. And so we opted to try and treat all of the water in the home, which we're only possible to do because Trojan donated all our devices. So we have a whole home device. We also have a large sample size. We powered our study to be larger than some of the previous epidemiological studies because they didn't see an effect of their intervention. And so we kind of maybe slightly overpowered it to 908 participants. We also have what I think is an innovative text message digital-based data collection system, which has yielded a greater than 98% response rate to date, which is pretty awesome. Historically, these studies were done with paper-based diaries that people had to fill out and mail in. And so I think we're making it easy for households to participate. And then we have this kind of innovative calendar option that when people identify like what days their kids were sick, it populates a calendar to say, okay, from this day that your child was sick, think back 7 days before and tell us about what happened in those days. And then they answer questions. And so we're hoping to reduce what we call recall bias through this data collection method. Some of our limitations is that we're relying on this self-reported illness data from the household. So it's only as good as what the parents can remember. And so there's definitely limitations to that. We only have a subset of samples being collected because of budgetary constraints. And when we've really struggled with recruitment because we started the study in the middle of a pandemic, which is really difficult with parents who work from home with small children. So we're trying to target the most challenging people in the world, I think, to get them to want to be part of the study. So that's definitely been a struggle, and we might have a hard time reaching our sample size. So I'll walk you through our timeline. So I received money from the National Institutes of Health in 2018 to fund a trial planning grant. So the trial is so complicated that they actually gave us money just to plan it, which was pretty great. And then we received money from the state of Pennsylvania to fund our pilot trial that just finished in the spring of 2022. And then while we were in the middle of our pilot, we started our NIH trial because we got that funded surprisingly early, and started that in 2021. And it's ongoing until we'll probably recruit the last people in early 2025. So what do I have for results? I don't have lots of exciting things to tell you because the trial is still going, but I'll show you some of our pilot trial results so far. And so this is a picture of the ultrafilter in case some of you guys don't see, but we have a little flow meter and the water gets hooked up outside and it's really contactless, which is great for COVID. So we don't usually have to go into people's homes and we can collect samples outside of their home. So preliminary results, we had 28 children enrolled in our pilot. Half of them were female. The median age was 25 months on enrollment. All participants were followed for 12 months and we had 0% dropout and a 98.4% response rate to the text messages. The gastrointestinal data we have, so we haven't unblinded ourselves and done full data analysis, so I can only tell you what the cases were across both groups. So in the pilot study, approximately 0.64 cases per child is what we saw of gastrointestinal illness, which is really low. For this age group, we would expect closer to 1.5 cases. So this is what we're seeing in the full trial, which is good that the illnesses have gone up, because otherwise our intervention would have an issue detecting an effect if it stayed at this really low level. But you can see that COVID and those restrictions really limited illnesses during that time period. We've collected, we collected 95 stool samples. 62 were in the absence of symptoms, 12 were in the presence of GI symptoms, and 12 were in the presence of acute respiratory illness symptoms. And there was no trend as to what we found and when we found it, depending on symptoms, which was really interesting. We're still kind of trying to figure out why, but we found adenovirus in about 15.8% of the samples, and most of those samples were actually in the absence of any symptoms. Norovirus G2, we found Gigatoxin 1 gene and enteropathogenic E. coli. I know those were found in asymptomatic children, which was interesting, and Cryptosporidium as well. Then for the water samples, the fecal indicator organisms, I'll talk about, we had really high contamination rates in these wells. Total coliforms, if you look at a per well basis, because we would collect repeat samples, 96% of them were positive for total coliforms. And for those who aren't that familiar, you're not supposed to have more than one total coliform per 100 mils. And so these results are, they're pretty high. E. coli, 46%, and Enterococci was 29%. And these are kind of the range of concentrations that we observed. And then highlight the E. coli, that's quite high. That's higher than what we're seeing in our full, but not, it's not in Ontario. I was comparing results of different provinces to the states, and in general, Pennsylvania has really high well water contamination. The rate of positive samples in Ontario isn't nearly as high for E. coli in particular. For our ultrafilter samples, we had 93 ultrafilter samples that were analyzed, and we detected Salmonella, Norovirus G2, Rotavirus, Cryptosporidium, and human Bacteroides in these samples. And then, so in general, between 13 and 27% of our samples were positive for human fecal markers. So that's what this HUM-M2 and HF183, meaning they were impacted by human sewage. 27% were positive for Cryptosporidium, 13% for rotavirus A, 6% for norovirus G2, and 4% for Salmonella. So from our pilot, what have we learned so far? Well, we've learned that human fecal material is getting into these wells, but we don't know what the burden of disease is attributable to drinking untreated well water, because the study isn't powered that way, and we haven't analyzed those results yet. We're still teasing through our illness data. The next steps are to finish the analysis. So we haven't done the saliva swabs yet, and we haven't done the comparison of the illness rates between the two groups. And also we're looking at how effective our blinding was. So we have a blinding index to look to see if all our participants were in fact actually blinded by the intervention. And our full trial is ongoing, won't finish till 2026. And we've enrolled 259 of our roughly 900 participants so far. And I'm pleased to say we've had one person drop out. So we might actually not need to get to our full 900, because we thought at least 14% of the people would drop out of our study. So I'm optimistic that we'll reach our sample size needed to execute the full trial. And so with that, this work is only possible with an army of people. I'd really like to acknowledge Dr. Debbie Lee. She was a postdoctoral fellow with me for 4 years and now is a faculty member at, where is she, Colorado State. And she was my trial manager, and now Jeffrey Turner is my trial manager. These are some of my co-investigators, and Chris and Peter are full-time research staff that work on this project. Joel, Aaron are at the USGS and USDA in Wisconsin that do all the sample processing. And then I have an army of other staff and undergraduate students that work in the lab and help tirelessly with recruitment and mailing out postcards by hand and doing a lot of like really tedious work. And I'm grateful for all of them. We also have an advisory panel of experts from across the US that we lean on for advice who have conducted these studies before. And I'd like to acknowledge all of them here. And then lastly, the funding from the National Institutes of Health and the Pennsylvania Department of Health and Trojan UV. And last but not least, all of the families that put up with us bothering them, and they all are so busy with lots of young kids. So we really appreciate all of their support. And with that, I'll end with questions.